and next presenter will cover the virus prevention and treatment and therapeutics. The next speaker is Professor George Gao. He is currently serving as the Director General of China's Center for Disease Control and Prevention. So when it comes to prevention and control of COVID-19, he is at the forefront. And also, he is former Secretary General of AFOB. Currently, Chinese Academy of Science, Institute Microbiology, and here he is working as a professor and at the same time Secretary General. Jiangxi Agricultural University, he earned BS degrees and also he earned MS degrees as well. And at Oxford University, he earned PhD degrees and he served as officers in China. The theme of his presentation is COVID-19, the virus, and prophylaxis and therapeutics.
Hi, everybody. This is George Gao from the Institute of Microbiology, Chinese Academy of Sciences, and also the Chinese Center for Disease Control and Prevention. What I'm going to talk about, the title is COVID-19. I'm going to talk a little bit about the virus and then focusing on prophylaxis and therapeutics, because this is really relevant to the biotechnology and biotechnic progress. So have a look at this slide modified from Dr. Tony Fauci. So we are facing a very serious emerging, re-emerging infectious disease challenge here. So you can see from this slide, we are encountering many viruses and bacteria, and also sometimes with some parasites. So many pathogens are challenging us. Look at it, this is what we have for the COVID-19. So we have more than 20 million confirmed cases and more than 0.8 almost million deaths so far. So it's really a serious problem, and we never thought this is a virus. We are expecting, or we know we might encounter coronavirus pandemic, but we never thought the virus, such a crazy virus, would be here. So this is what we are having at the moment. First, I will give you a very brief outline about what happened in China and what we have done, especially for the prevention and control, and what we have done, what we've been doing at the moment for prevention and control for such a devastating virus and COVID-19. So this slide tells you the timeline and what was going on. Retrospectively, we found in early December, we already have some unknown pneumonia with unknown etiology, PUE. It's already there. But, we, by early January and the end of December, so roughly we sequenced the samples early January, we already know what it is, and we isolate the virus as well. So we have the virus, we have the genome, it's a novel coronavirus. And then on the 23rd January, and Wuhan, the original place where the virus first identified, were closed down, were locked down. So the containment started there. You can see from this curve, very rapidly, the disease is under control in China. Eventually, on the April 8th, and we claim Wuhan is free of the COVID-19. So that takes about 3 months there. So this, look at what happened in China. So we did a great job. Of course, so everything's under control. Think about in January and February, and we have so many cases in Wuhan. In total, we are talking about 90,000 in general, 80,000 in Wuhan. And also, we are still doing a lot analysis for the antibody positive serology, and we will know the answer soon. So see how many asymptomatic infections there. Look at what happened since starting from April, May, and especially something in June in Beijing, and then some more cases in Dalian. We have a Dalian outbreak, and here you have a Xinjiang outbreak. You can see, compare with what happened in Wuhan. And whatever you saw recently in those regions is relatively very small numbers. And we learned a lesson, and we know how to get it done. And obviously, the disease is well controlled in China. So have a look in Wuhan. This is in Wuhan. And the red one tells you the imported cases. When the disease was got it down, and we have very small numbers of local cases, and all the others is from imported cases here. And of course, in June and July, in Xinjiang, Dalian, and Beijing, they are initially imported cases. But of course, we have some localized cases eventually. So this is what's going on in China at the moment. So this is to give you an enlarged picture to see what happened there for the recent from March to June and July. These are very small waves in China. So now I will come to tell you about what we have done for the virus discovery and identification. So this is the virus we isolated in January. On January 7th, we saw the virus under the EM, and we got several groups working together to confirm. Because of the Koch postulate, we need to make sure this detect the virus with a causative agent. Several groups have the convergent results. It confirms, so this is the virus causing this disease. So this is the milestone, tells you, the whole timeline, what we have done, what we saw early in China, December 31st, and we got the samples, and January through until January 29th, we reported everything on the journal called New England Journal of Medicine. And also on January 24th, the initial report for clinical observation, it's in the Lancet. So they all reported there. And of course, after we found the virus, and we shared all this data with everybody in the world. Of course, after that, the diagnostic kits was developed, and it has been used for a while. So this is what we saw here. And the sequencing sharing within the GISAT, and also in China CDC weekly. This is the journal, official journal from China CDC. So what it looks like. So the virus is a novel coronavirus. It's closely related to the virus isolated from or sequenced from the bat. You can see from here, SARS coronavirus, and this is a coronavirus now it's called SARS coronavirus 2, and also it could also be called COVID-19 virus. We call this a novel coronavirus 2019. So this is the virus. So this is the paper published in the Lancet from our group. Of course, after that, it confirmed the virus can be found in the stool samples of the, that tells you the virus transmitted by droplet, and the virus can be found also in a dissective system, at least in the stool samples. Of course, I mentioned on the January 29th, all these preliminary epidemiology and transmission dynamics was reported in New England Journal of Medicine. You can go and read this paper if you have interest for the detailed description. And China CDC Weekly have a second report for the 17th February about some more cases for the first wave what we saw in Wuhan. Now I will share with you what we have done, what we are doing at the moment in China for the strategy of the COVID-19 control and prevention. First, when you think about at the moment in the whole world, we have the prevention and control strategies with different one. In China, in Wuhan, we use the containment or lockdown strategy. Of course, look at this projector and here, and the purple line tells you if we haven't done so, i.e. if no intervention. Of course, there's no specific intervention. All the intervention is not specific. It's NPI, non-pharmacological intervention method. So if you don't have this one, the purple line tells you we would have so many cases, and you can see the epidemic peak was reduced in China due to the strategy we introduced. So this will give you, if you want to read further about what we have done in China, and all this data were presented in Clinical Infectious Diseases, the journal. We call it the first 100 days in China for the prevention and control. And go and read this in that journal. Of course, what we have done, I said NPI, non-pharmacological intervention. So it's really so-called traditional containment method. The containment strategy, including, of course, we call the three major stuff: mask, social distancing, and personal disinfection measures. Here, I want to give you mention a little bit about the whole list, what we have done here. You can say, of course, it's social distancing, detection, isolation, and quarantine, and movement restriction, including suspending public transport for a while in Wuhan, and also infection prevention control. Here, I want to address more on nosocomial infection, i.e., it's the hospital infection control, and also environmental measures and social mobilization. And here I want to address more about risk communication. Risk communication is very important, as while we are encountering the problem for the COVID-19 pandemic, we also have something because at the moment the social media, we have the so-called infodemic. The infodemic means it's information and misinformation. They can be zipped together, giving you the wrong information to the public. And then mention about personal protective measures, of course, hand hygiene, mask wearing, or mention about this. So this is a NPI, it's very important. So when in China, from the very beginning, as I said, those figures you saw here, they are presented in Clinical Infectious Diseases. It's called 100 Days of Coronavirus Disease 2019 Prevention Control in China. Go and read there. We call 4 lines, 4 levels, and 4 earlies. So the key term is, what is the 4 lines? 4 lines from the very beginning, we know it's a quite complicated case. So we got to have a very good refined strategy. So we introduced this like the first line would be Wuhan and Hubei. That's where Wuhan city is located in Hubei province. And then the second line is our capital. You got to protect your capital to make sure the public understand what you are doing and make sure the public has no fears. So this is why I put a lot of effort for the second line in Beijing. And the third line is in the Hubei province and Hubei province surrounding regions, and including the neighboring provinces. Of course, the fourth line is nationwide. Those defense lines, 4 lines, we use different strategies and use different human resources and of course, logistics. They are so different. Go and read our 100 days paper. Of course, for early, it's very important to early detect of the virus and also early report to the authority, early report, alert to the public. Of course, early isolation. Whenever you found cases, you got to have the case isolated. Of course, if you have a severe cases or it's a symptomatic cases, you got to have them treated as early as possible. So this is the wall. I use Abraham Lincoln's Gettysburg Address to say, this is the wall. It's for the people, by the people, of the people. So this is why we really address from the very beginning for the community engagement, for the community health and education, for the community's hygiene and disinfection. So you got to get the public activated. They know what they will do. They know what they should do. They understood what you are doing. They understood what they should do. Those are very important. Of course, 3C is also very important. Coordination, cooperation, communication. With such a big disaster, resources allocation is very important, including healthcare workers and PPE, all these logistics support, and plus, public understanding and the epidemic response, all this, you need everybody to work together. So coordination, and you need everybody cooperate together. Of course, from time to time, you need to communicate with the public and with the authorities. So after we got the virus down in Wuhan, we in China, we moved to a new stage. I call it dance with the virus. In the end of March and early April, we try to strictly prevent importation and community resurgence. Here, we want to say, from the very beginning, imported cases control is very important because by April 4th, we didn't have any more cases in China. And you have to have a refined measures. So we call it tailored control measures at varied risk levels. And for early measures in the new situation, strengthen case reporting and surveillance, enhance laboratory test capability in country level, not just in the local level, and also maintain graded community prevention and control measures. And we gradually resuming our production and work. The society really restart a lot with the new, we call it new normal. Of course, and prevention control protocols at varied risk levels, like close contact and cases, and general population, and of course, severe cases and asymptomatic cases. You have to try to define all those cases into different and varied risk levels. And of course, from the very beginning and until now, we always prepared ourselves to make sure wherever we encounter new severe cases or severe scenario, we have to get ourselves prepared. So that is why we call it improving emergency management mechanisms and operational modes, and disease surveillance epidemiology capacities, service capacity of medical systems, maintain social functioning and order, protect vulnerable and high-risk populations. You have to make sure those who are vulnerable, they got the proper health care. Of course, OPI, non-pharmacological interventions. And meanwhile, I will get into detail about the vaccine and drugs later on. So from the very beginning, we tried to use some approved drugs, and also we also try to develop something by using the traditional method and also the 21st century, try to develop the vaccines. I'll come to that point later. Now, after the Wuhan and also the Northeast region, there we had some imported cases from Russia. And in mid-June, suddenly we have something from Beijing. So I call that one the whack-a-mole. So you found something, you get suppress them. So there, from Beijing's response, which we didn't react properly, it could be a second Wuhan. But we are lucky enough, we work so hard by doing those I want to read here to share with you. Restriction instead of lockdown. We didn't have a lockdown in Beijing. Risk assessment based in a low risk, middle risk, and high risk. Of course, we have a very strict rule for the 14-day quarantine for people coming from Beijing. Anyone, because we have Beijing, we have something in Beijing, anyone want to go out from Beijing. So we have 14-day quarantine. That's very strict. And quickly expanded PCR testing. We did a high-risk population. We did a very high load work for the PCR-based nucleic acid testing. So we did what we have done. 2.3 million persons in 1 week is done in Beijing. So that's very important to get anyone who might be a risky source of infections. Of course, secure economic resuming, restriction in community and district level, maintain the statutory factory running, resuming. So that's important. We want to maintain new normal social life and efficient whole society response mechanisms, including national and local government involvement, society, community level involvement, and NGO and private sector's involvement. Of course, the key to get the individual level prepared and mind prepared. Of course, if you want to read something about what we have done in Wuhan, and also we try to share with everybody in Beijing, now in Dalian and Xinjiang, we published this paper as health policy in the Lancet called Active Case Finding with Case Management, the key to tackling the COVID-19 pandemic. The community level case finding is the key. So you have to do this actively. So this is very important. Exactly what we have, that's just after Wuhan cases. We thought this is important. So we just follow this rule. We had done a good job in Beijing, Xinjiang, and Dalian, of course, also Northeast region, including Harbin and Sulan, and in Jilin province and Heilongjiang province. So this is what we have done. It's a very good job. Of course, and you have to compare COVID-19, SARS, and 2009 pandemic H1N1. You can see, again, COVID-19, the case facility rate, safer could be really varied. So why it's varied from the very beginning? And people got panic, and they are really worried. And so this is why you got the safer like 5.9. Like SARS, 10%, and the pandemic is 0.0 to 0.4, and seasonal flu 0.1. This is the case fatality ratio. But once we know what it is, what we have done in Beijing, starting mid-June, June 10th, the first case. But look at the first reported case, and we didn't have any death case this time. We learned a lesson. We know how to treat the patient. We know how to trace back to any suspected cases and any close contact. We didn't have any death case. So this is where we are now. Of course, when we go and read our paper, we call it the different strategies: containment, suppression, mitigation. At the moment in China, we didn't do any containment. But, however, I want to remind you, the containment measure or lockdown in Wuhan from the very beginning, played a very, very important for China. Otherwise, China is such a big country with 1.4 billion personnel here. So it could be a very serious problem. So we did a containment in Wuhan, but when we found something in eastern provinces and also Beijing, Dalian, and Urumqi, we didn't do that. We only use the suppression and mitigation. So, you got to think hard and to implement different strategies with different cases there. So, this is a very, very important strategy for you to follow. So, core measures for the containment and suppression. You can see the case detection, contact tracing, isolated case management, those three are very important factors for you to, gain by using the suppression strategy. Now I want to come to the science part. So I talked about the background, what we have done, what we found, and then I came to the strategies and disease control prevention. Now I want to come to the science part. As a scientist, I give you a little bit about what I thought and what the world have done, and also the work, especially in my lab and my collaborators' lab, what we have done for this COVID-19. First, COVID-19 virus entry. I mentioned the COVID-19 with several groups, 4 or 5 groups from the very beginning. So then we came to the same conclusion. It's a novel lineage B beta coronavirus. So that's very, very important from the very beginning with different groups. We didn't want to make any mistake. We want to make sure whatever we found is genuine. It's from all the labs we got the same results. So this is something from my lab. We also from the very beginning we confirmed. Look at those pictures, and this is the binding, soluble protein binding. We confirmed the spike protein receptor binding domain in the C terminal of the S1 bind to the hormone ACE2, which is also the receptor for SARS virus. So you can see, all these tells you the same result. We did the crystal structure. 3 more groups did the same thing. It's come to the same conclusion. So this is the paper we published in Cell. Tells you how COVID-19 virus bind to the hormone receptor ACE2 through the receptor binding domain, or it's also called C-terminal domain of the S1 protein of spike protein. Now I want to come to the real biotechnology part, to the rational vaccine design and where we are for the whole world for the vaccine. So this is the coronavirus. You can see it's on the surface looks like they have a crown. So this is why it's called coronavirus. For the vaccine at the moment, first, of course, this is the traditional method, inactivated vaccine, i.e., you grew the virus and by use chemicals, you inactivate the virus by adding the adjuvant, mix them together, and then you have a inactivated vaccine. So this is one of the lines for the vaccine development at the moment in China, and also it's in phase 3 clinical trials. Looks like it works very well. Of course, this is the technology you have been used from the very beginning, of the 20th century, and even the end of the 19th century, people start to use the inactivated vaccine strategy. And now live attenuated vaccines, and a lot of groups, they are doing the passage of the virus to try to get some attenuated virus. If you can get an attenuated virus, so that virus will stimulate the human body immune response, but without any pathogenicity. So this is ideal. But at the moment, no one ever got a live attenuated vaccine yet. So, so many groups in the whole world working on it. And the third vaccine, also this one, we will finish up the phase 2 clinical trials in China soon. It's a protein subunit vaccine. So this is developed by my group. And you can see, and the ACE protein forms a trimer, it's a trimeric protein. And here, those egg-like stuff here, it's a receptor binding domain. So this is a monomer. Receptor binding domain, we already confirmed it's give you a very strong immune response. So can we use this one as a vaccine? So this is actually what we have done. It's a protein subunit vaccine. It's a dimer. And viral vector vaccine, you heard about Oxford chimpanzee adenovirus 26. You heard about human adenovirus 5 vector in China. A lot of viral vector vaccines here, is under development. The last one, nucleic acid vaccine. The mRNA vaccine, you heard something about the Moderna vaccine from the US is under clinical trials. I think they are also approaching for the phase 3 clinical trials. Of course, when you are talking about the nucleic acid, you are also expecting the DNA vaccine as well. So here are the vaccines under development in the whole world, and I want to give you a very quick example about the science part of the 2 vaccine I am involved. First, inactive vaccine is called BBIBP CoV. So this is the vaccine, and for the decryption of this vaccine, the development of the virus selection is just published in Cell. And before I'm talking about this, I want to tell you, people keep asking, can we really have a vaccine for the COVID-19? Have we learned anything from the past? Here, I give you some example. For the human being, we never use a coronavirus vaccine, but some animal viruses, including the pig, TGEV, and the pig, PEDV, and the cat, PIFA. So we have very good vaccine, are being used for the animals. So that tells you we are practically in a very good position. We might be able to get a very good vaccine because those vaccine works for the animals. So this is why even Dr. Tony Fauci and I, we believe we are cautiously optimistic for the vaccine development because we learn from the animal coronavirus vaccines. And this is tells you the inactive vaccines. They are under phase 3 clinical trials and also some in phase 1 and 2. So this is in China. You heard about it. And also in India. They develop an inactive vaccine. At the moment, only 2 countries in the world, China and India, we have the inactivated vaccines. And for development of the inactive vaccines, it's very important for you to have a seed virus. So selection of a seed virus is very important. This work done in China CDC set a good example. We compare with a different virus. So this is the one, HPL2 is a very good one. We selected this virus, and this virus being used later as a seed virus for the inactivated vaccine development. So how we develop this? So cell recovery, cell factory for the fermentation of the cells, and then bioreactor. Of course, we inactivated the virus by using the beta propyolactone, and it works well. And after this, we purify by buffer exchange, and of course by the chromatography, and by adding the adjuvant. For the adjuvant, we use aluminum. So we use aluminum as an adjuvant. So then we have a vaccine. So this is the really very, very traditional protocol here. Because we did, before we got to the human trials, we put this one in, the neutralizing antibodies in the rabbits, guinea pigs, rats, mice, and monkeys too. You can see, even one dose, you got a very good immune response with different doses. And of course, if you have 3 doses, you get a better one. Of course, I want to tell you, even 1 dose, you can get a very good response. At the moment, we are doing 2 dose or 3 dose clinical trials. I'm pretty confident, 2 dose for most of the people should work well. So protective efficacy of this virus in monkeys. This tells you, we inactivate for day 0 and day 40 the second immunization. And we did this in the monkey. You can see neutralizing antibody titer grows, and we try to do for the throat swabs, you can see down, and this is the control. So that tells you, and also lung lobes for the virus. Obviously, you can see with this vaccination for the monkeys, and the monkeys can resist for the challenge, from the, when you did this virus titration in throat swabs, anal swabs, and lung lobes. We got very good results here. Of course, look at this placebo, very clear lung inflammation, but you didn't have something here for the vaccinated group. Of course, you might have a little bit, but in general, it's protective. And then we started for the clinical trials. So this is we started for the clinical trial. This is the timeline for the whole United vaccine development. So this is at the moment this vaccine is already in phase 3 clinical trials. Now come to the protein subunit vaccines. So the work I'm involved. So this is I encourage you to go and read our paper from my group in Cell. So this is the abstract, graphical, physical abstract to tell you, this is a virus, this is a trimeric S protein, this is a monomer, and this is a receptor binding domain. What we by accident actually, by chance, we found a dimeric receptor binding domain linked by a diaphragm bond, and then we developed this one by tandem repeat to link them together. And this strategy was first initially used for MERS, and then we found it works for MERS. Once we get the COVID-19, immediately we tried to follow COVID-19. Again, it works very well. And we prepared the mammalian cells with this strategy. We got a very good expression. This is a vaccine under the phase 2 clinical trials at the moment. So this, again, same thing. I don't want to go in detail because of the limitation of my talk's time. I want to tell you same thing what we have done here. Different animal challenge and also tells you about challenging the monkeys. It works very well for the this vaccine. This tells you, the dimer. We also did the crystal structure. We saw the structure to tell you what they looks like. So this is really, it's the dimeric receptor binding domain with a disulfide bond or without disulfide bond. We got them to linked together. Of course, we did the work with this vaccine. It works in the ACE2 transgenic mice, and we can see this, again, placebo vaccine, and the enlarged, you can see, it's protective, the interstitial pneumonia in the vaccine group. It works very well. Of course, this tells you it's under clinical trials. Now we are in the phase 2. Now, potential therapeutic antibodies. So, what we did from the infected patients, we got the isolate memory B cells, and this is just routine work. Isolate the virus, pair the heavy chain and light chain together. What we got several monoclonal antibodies. One of them we call CB6. It works very well, bind very well, and this is the one. We did the one for the in the UV for cell-based experiment. Very good neutralization efficacy here. Of course, we got a crystal structure as well. We saw the structure to see, how the antibody and bind to the receptor binding domain of the S protein. Of course, we also introduce LALA to eliminate the potential ADE effect. And again, we use the monkey to test. We got a very good protective effect here. So the paper published in Nature. Go and read this paper. Of course, because you all have once you use a neutralizing antibody, you might have some antibody escape mutation. So we really want to have pair of the neutralizing antibodies. And then here I want to test you. And then we got a pair of B38 and H4. We got 2 monoclonal antibodies. This is how they bind to each other. And H4 and B38. So we mix them together as a cocktail. So this cocktail works very well. The paper published in Science. I encourage you to go and read the Science paper. Of course, this is already under clinical trials by collaboration with Eli Lilly and Junshi. We are working together. Now, quickly about after that, my group also tried to use the HCV inhibitor, protease inhibitor, or boceprevir, and it works. This is the main proteins from the COVID-19 virus. So, and what we have done, we screened all these known HIV, HCV protease inhibitor. We tried very hard. Finally, we got two of them works very well. One is Perceptrovir, the other one is GC376. It works very well. We got the crystal structure and to show how they bind to the enzyme, and it works very well. This is a detailed binding for the GC376, and it works very well, and also works combined together. If you work together with the remdesivir, they have some collaborative effect. Works very well. I think so. Here is what we have done. Outline for the outstanding question. First, correlates the neutralizing antibodies with protection. We have the neutralizing antibody. Can those antibodies would protect the infection? And can the neutralizing antibody can really reach the lung? And also, how long the neutralizing antibody would last? There's all these questions asked. Can we really would have the ADE, for the vaccination? And also, we need to have a very good immune programs and make sure we have very good results. Thank you very much.